Right. So this next talk puts me in mind of one of Saint Augustine's writings, who said it is not less noble to study worms than to study stars. But in fact, the theme of this talk tells us that actually by studying worms, you may actually learn something about life out among the stars. And the talk is by Netta Cohen at the University of Leeds. What makes life successful? Lessons from a worm.
Thanks very much. This is super exciting to be here. I know I know nothing about extraterrestrial life, but let's go back to very, very basic principles and ask from a whole organism level, what is an organism? And as the word suggests, and etymologically that is the source, it's presumably something that organizes or has some internal organization. So what does that tell us? What could that be? Well, when we think about organization, certainly as physicists, we might think about a solid with some crystalline structure. Living matter is nothing like that. It's irregular, soft, gooey, full of fluid, as we've just heard. And so the word organization might be referring to the large-scale organization at the organismal level, the modularity of the structure on the inside, or over time, the coordination of action of living matter. And what is the implication of such organization? Well, a system has organization suggests that it has order, it has a low entropic state, not so low as a crystalline solid, but much, much lower than random, so that it can store sufficient information. And this happens without an external organizer. So it's autonomous, it self-organizes, and this requires an active entropy reduction mechanism that to self-organize the system and even to self-repair it. And in order to maintain that low entropic state, living matter is obviously out of equilibrium, far out of equilibrium, and it absorbs fuel and energy, including chips, from the environment to maintain its internal order and this low entropic state. And if it loses that, we die. And so that's not the only energy we absorb, not just in order to maintain that low entropic state, but additional energy is absorbed in order to allow that organism to act on the environment in a meaningful way, in a beneficial way, which we usually interpret in an evolutionary context. So armed with that intuition, what can we say about what that something is that organizes or has this internal organization? What could such somethings be? And to address that, we can go back to three of the greats. Sir Erwin Schrödinger, What is Life? Son already mentioned, basically premised all of his studies on cellular life, the processes within the cell and how those cells form multicellular organisms from a physics perspective. But and that's not surprising. That's because, of course, we have all life as we know it today on Earth has evolved from some common ancestor, which was some sort of a protocell. But there are other approaches. So John von Neumann was looking at the feature of self-replication. And so he didn't necessarily have a compartmentalized cell. He still had modularity and specialization, but he was thinking, what are the components that are needed and how must they interact for a machine to self-replicate and design this Gedanken machine? And Freeman Dyson, decades later, approached the question from a slightly different perspective of asking, what are these macromolecules that can self-replicate, that could have self-replicated before there were cells? And that has introduced this tension in the history of the origins of life on Earth about what might have come first, the cell or the compartment or these self-replicating macromolecules. We don't really know which came first. We don't know anything about what happened before, or almost nothing about what happened before, and very little about what's led from there to the richness of life as we know it today. But once you have a definition of a cell or a compartment, that defines an inside and an outside. And you can also generalize that to multicellular organisms that have an inside, a well-defined inside and a well-defined outside. And that gives you identity. And that identity also has a certain time scale, which we now can associate either with a cell division or with the generation time or even with life and death of an organism. And we still have those functional properties that we talked about earlier: self-assembly, self-organization, development, self-repair, self-replication, and so on. And all of those somehow subject to the rules of evolution now that we have cellular definition of inside and outside. But all of those definitions require that compartment, that cell. Now, we do have acellular life on Earth, even to this day, including viruses and prions, but those do need a host. They need cells in order to replicate. And so we don't really know what life would have been like had we not had cells, had that first protocell not developed as we know it. And so going back to that question of modularity, with or without these compartments, we have extrapolated that idea of modularity to all complex life or features of life as we know it. And in the context of animals, we think about separate sensors and separate motors. So we have sensory organs and we have muscles and so on. And so just to try and veer you gradually towards the worm, let me ask a riddle. How many animals do you know that respond to colors but have no eyes, or that respond to sound, even directionally, but have no ears? And the audience in this room might not know of many because we think about big animals, but actually this is not uncommon among very small, compact animals. And the worm is one of them. And so it has a complex life cycle. It's free living. It has lots of complex behaviors, all with under 1,000 cells and only 302 brain cells. And for those of you who have not yet spotted it, here it is to the scale of an old 1-pound coin. It has all of these behaviors, and it does that with this very, very, very tiny brain, which has been fully mapped, all 302 neurons and their connections. Now, obviously, it does not play chess, but it does everything else that a free-living organism has to do. And that includes lots of sensory modalities and lots of motor actions. And most of these sensory modalities do not have distinct sensory organs. So smell and taste does, but all of these others, touch and osmolarity and electric field and magnetic field and color and sound and others that we probably haven't discovered, we don't think about as having been based in a sensory organ. And so the question is, how does that system organize, modularity or not, to generate these complex behaviors? And what can we learn from such a simple animal about the very principles, basic principles of life on Earth, and can we break them to think about more general life? And so let's start really simple. Let's just think about the shape that life takes. And so everything from viruses and bacteria all the way up to animals through fungi and plants and all the rest of it takes on various forms and shapes. But one thing that we notice by looking at those is that either the organism as a whole or parts of it contain long and thin parts. And that is interesting. And obviously it's true for a worm. And across scale, we might suspect that this is not an accident, because things that are long and thin have advantages across all scales, from molecules to large life forms. They're flexible. They support axial polarity, so head and tail. They're great for motility in lots of different environments, water, surfaces, underground. They support lots of manipulation and motor activity, from burrowing, reaching, gripping, latching on, jumping, and leaping. They're very compact on the inside, and that supports energy transport, both passive and active, and material transport, and they recur as modular structures again and again in life forms as we know them. Let's go back to this notion of modularity. When we usually think about modularity, we think about the brain. And so you might think, well, with 302 neurons, how much modularity can you have? But if you look at the concentration of sort of the analog of white matter of our brains in the worm, then you can see that it actually is modular, with some 5 very clear bundles of nerves that each support its own compartmentalized or modular neural circuit. And you can use that bundle organization to try and follow the flow of information from the sensors out to the behavior of the animal. And what you find interestingly is that if you start with the sensors at the top and follow each of these bundles, at the bottom, before you actually generate the behavior, you have this massive recurrent layer with lots and lots of communication and coordination and integration of internal state and all of these different circuits in order to generate coherent behavior that makes sense for the entire organism. And so on the one hand, modularity allows us specialization and interaction between modules, but on the other hand, to act appropriately, complex life must integrate over the modules to coordinate the output. Okay, and so how does that happen? With 302 cells, how do you generate coordinated behavior? Well, it turns out that you don't have many cells, but you need to generate lots of versatile, very rich behaviors. The animal moves differently in different media and even in 3D, and it does that with incredibly compact circuits. And the way that it does that is that the control relies on the physics to produce the robustness and the versatility and the adaptive behavior. So here's an example of a really simple circuit that's sufficient to generate all the forward locomotion. And you can now combine that with backward locomotion and with turning and so on to try and understand how the animal forages efficiently in 3D space. So here it is moving around in 3D space. So I've already mentioned that even though the system is highly modular, the simplistic distinction between sensory organs and motor action is not that straightforward. So let me give you two more concrete examples. So the first one is the same locomotion example that I gave you before. In order to exploit the physics and bring it into the motor action, what these motor neurons are doing that actually generate the activity is they themselves sense the shape of the body. So each cell is actually a sensory motor loop within this motor circuit. And the second example is here. This is how the animal forages around for food by following chemical gradients. And these chemicals may or may not indicate the possibility of the presence of food. And so the animal needs to search these many, many different patches. But rather than having sensors that encode the environment, then transmitting that to some complicated brain that makes magical decisions about where to go and then going there, it's the sensors themselves that are turning on and off to modulate the attention of the animal and to actually balance between the exploration and exploitation. So you can look at the activity of the sensory cells and already tell whether the animal is in a sensor, in an exploratory mode, or in an exploitatory mode, and exactly which motor action it's about to take. Okay, and this first of all brings out the notion that for the animal, there isn't an objective value to the many, many cues that it's sensing from the environment, but rather these cues are relative, and the animal is constantly assigning subjective value to what it's sensing in order to make the right decisions. And this is also true in this example, which brings out another aspect which I think is pretty universal in life, which is risk aversion. So when the animal is deciding whether to do something or not, it's not weighing up the cost and benefit of a specific cue in isolation, but it's actually realizing that this is within the context of its whole life, maybe not in a self-aware way, and death has an incredibly almost infinite, if not infinite, penalty for that individual. And so therefore, it's going to be risk averse. And so this is an example, both experimental and simulations, of animals surrounded by a sort of ring of fire, in this case, fructose. And under normal circumstances, they don't want to cross that ring of fire, even if there's a smell of food on the outside. But when they're sufficiently hungry, well, boy, they will cross it. And so this sort of closed loop control that allows you for this dynamic action and assessment of the different cues of the environment is very different than rule-based logic that we find in many other systems and models of intelligence. So everything is relative to the animal. Before I finish, I wanted to give a shout out to work that is not done in my lab. Two different projects, this one from the Kokabas lab about collective behavior. So if you put the animals a few centimeters away from food, you expect them to go to the food and eat it. But they don't actually do that. What they do is they approach the edge of the plane here, and then they form this superorganism, this collective 3D entity that then acts as a single unified system to go and eat the food all together. And then once they're done, they just disperse. And this idea of the individual basic compartment not necessarily being an organism, but being a superorganism, and the ability for these structures to be transient is really interesting. They can be variable size, they grow, they combine, they split up, and they disperse all together. But when they're together, they act as a single unit. And there's a second example. This one comes from the really interesting, resilient life cycle of the animal. So when the animals have plenty of food, they just reproduce and grow. But when food runs out, then the juveniles that hatch from the eggs are not able to develop as normal, and they form this highly persistent, energetically efficient dower state, dower larva. And these dowers, they're absolutely desperate to get somewhere with food. So they will form these immense towers consisting of thousands or maybe even millions of animals. And those superorganisms behave coherently as a single system. So they will just go and move around seamlessly, all looking to something to latch onto, or some sort of a pollinator. And if some of them are picked up and placed in a food, somehow arrive at an environment where food is, they will just disperse. So with that, I'd like to say, well, what have we actually learned from all this? Obviously, C. elegans is cellular life, because all we know of on Earth is cellular life. And that means that there is a separation from inside to outside that provides compartments. And these are the basic units of life that can specialize their function and work together. This is really all we know about on Earth. But we know more fundamentally that whatever the compartment, whether it's cellular or not, whether it's transient or fixed, it must maintain low entropic state. It must act strategically on the environment and be adaptable and robust and resilient to survive successfully. And so perhaps I can leave you with a question of, could life prosper with conditions weaker than some fixed compartmentalized? And what would that mean for what that life would look like? And also what would be its biosignatures? And with that, I'd like to just thank my amazing team and collaborators and the organizers of this really exciting meeting. So thank you.